All right. So again, I apologize for not being there on Friday this week, although I think that's amenable to most of you. Today will be the last discussion on really the organs specifically that are involved within the digestive system. On Monday after break, we're going to talk a little bit about digestive chemistry and a few other things on that following Wednesday, but this will be the last one to really talk about the structures themselves. So the digestive tract that we've been talking about up till now, I've mentioned these organs. These are called ancillary organs. So they are not strictly part of the digestive tract. Food is never at any point going to enter and be processed in by any of these organs. However, they provide some aid in the process of digestion. The three main organs that I'll be talking about are the liver, gallbladder, and the pancreas. The liver is a very large organ that sits on the upper right-hand side of your abdomen. It is actually attached to the underside of your diaphragm, so it does move as the diaphragm moves, which is a pretty interesting notion. And the liver is hard to summarize because it fulfills several very important roles to the body that are part of different systems. So we're going to focus on how it relates primarily to digestion, but we will also cover a little bit about how it's functioning for the processing of blood as well as vitamin storage. So the anatomy of the liver, it's fairly straightforward. It has two lobes, so similar to the lung and how the lungs have lobes, the liver also has lobes. We have the larger right lobe and the smaller left lobe. These are going to be separated by this structure you see right here called the falciform ligament. Falciform means sickle-shaped, and so this ligament happens to have a somewhat sickle-shaped structure to it. That's how it got its name. And this is the structure that connects the liver to the peritoneum. Recall that the peritoneum is that fluid-filled sac that fills the abdominal cavity and helps to support the organs within it. Now, notice I used the word ligament here. This is not strictly the same way in which we discuss ligaments when I was talking about bones and muscles. Ligaments, in that sense, connect bone to bone. But we use the same term when we're talking about how organs connect to each other. So the falciform ligament is connecting this organ to the peritoneal sac. The liver also has a really interesting role in fetal development, and we will come back to this in a little bit more detail when we go through human development later on in the semester. But I wanted to cover it here because it's part of the circulatory role that the liver plays. And it plays a different circulatory role during fetal circulation than it does in a fully grown adult. The fetus is not going to be breathing. The lungs are completely inoperable. The fetus does not have any exposure to air, so there's no way to get oxygen to the system through the normal circulatory system. Instead, oxygenated blood must be supplied by the mother via the umbilical cord from the placenta. So if we look at this figure, we have the placenta in the mother, we have the umbilical cord, which is containing a large artery, and that's going to be containing oxygenated blood, which will then pass into the liver of the fetus. So the blood comes in from the umbilical cord right here. And this umbilical cord, once it has entered the fetus, is now called the umbilical vein. The umbilical vein is carrying oxygenated blood, and it will meet up with the fetal liver. And from here, the blood can take one of two pathways. It can either go directly through the liver, or it will pass over the liver in a structure you see right here, and we call this the ductus venosus. So this is the pathway that blood can take that bypasses, just crosses over the liver. and then continues on into the inferior vena cava. Now, the inferior vena cava is normally the repository for all of the cleaned, deoxygenated blood from the lower half of the body. Well, that is still the way it's operating in a fetus, because the fetus is still processing blood throughout the limbs. So we're getting deoxygenated blood up from the lower half of the fetus, and that's going to mix then with the oxygenated blood coming from the mother. So the inferior vena cava is now actually a combination of partially deoxygenated and oxygenated blood. From this point, the blood is then going to pass into the heart, and it's going to bypass the normal circulatory system that we've discussed for heart function. So the inferior vena cava sends this mixed blood into the right atrium, and there's no point for that blood to enter into the right ventricle and be shunted out through the pulmonary arteries into the lungs, because the lungs are not operating. There's no point, there's no oxygen that's going to be added at this stage. So instead, there is a hole that exists between the two atria. It's called the foramen ovale. I mentioned that earlier on in the semester when we talked about heart defects. So it passes through the foramen ovale into the left atrium, passes down into the left ventricle, and then out through the aorta in a normal pathway for blood into the circulatory system. After birth, there are going to be many changes to the circulation, some of which we've talked about and some that we'll come back to when we look at development later on in the semester. But because the lungs are working, we're going to change where oxygenated or deoxygenated blood is going. One of the changes involves the liver. That ductus venosus, that pathway that blood could take that goes across the top of the liver, is no longer needed because the infant is no longer receiving blood from the mother. Instead, that vascular structure is going to wither and actually becomes an elongated ligament. And we call this the ligamentum teres. And it's something that you can actually see if you've ever looked at a liver of any kind of mammal, or if you have participated in a human dissection. You can see this large flap-like structure that hangs off the front of the liver, and just a remnant of how your mother passed oxygen into your system during the fetal stages. All right, so now we're going to talk about how blood is passed through the liver. Most of the materials that are going to either enter or exit the liver are coming through a single spot, and we call this the portal hepatis. The portal hepatis sits on the underside of the liver, so you can see it kind of in this figure right here. And this is where most of the ducts and vasculature are going to be entering or exiting. The contents of the porta hepatis is called the portal triad because there are three, tri meaning three, major vessels that are going to be passing through this opening. The first is the hepatic artery proper. This is carrying oxygenated blood, so you can see it right here, hepatic artery. And it's carrying the oxygenated blood to the liver as its own supply of blood. So the blood that the liver needs to survive on its own, not the blood that it's filtering. The hepatic portal vein, one we see right here, is carrying all of that dirty, deoxygenated blood from the lower half of the body, and it's going to be filtered inside the liver. And then finally, the third portion of the portal triad is the common bile duct, which is how we take the bile produced from the liver as well as the gallbladder and send it out of the liver into the digestive tract. So we have two objects that are entering material and one that is exiting material. The exception to the porta hepatis is instead the hepatic veins. So this is how we take blood from the liver that has been filtered and cleaned and exit it into the inferior vena cava. So the hepatic veins are typically going to leave either from the top or partially on the top back portion of the liver, empties all of the cleaned blood into the inferior vena cava, which will then send it back to the heart into our normal circulatory system. As I said, one of the liver's primary jobs is going to be detoxification of blood, and it's going to be using that portal venous system. Excuse me, this is not my normal computer. So all of the blood that's being retrieved from the small intestine, from the large intestine, from the rectum, the duodenum, all of these structures, including the spleen over here, all of that blood is coming back and shunted through the hepatic portal vein, through the porta hepatis, into the liver, where it will be detoxified. The liver also serves some important roles in the form of metabolism of different nutrients, as well as the breakdown of structures for digestion. So things like carbohydrates, lipids, and proteins are a little bit harder to digest directly, and so we need some assistance in that process, and that's going to be through the production of bile. So while the blood is being filtered by the liver, it's taking out those toxins and it's breaking them down and producing this material we call bile, which we'll talk about here in a minute, but it is used to break down particularly the lipid structures. The liver also acts as a storage location for glycogen. You may recall glycogen from last semester. We talked about this molecule. It's like polysaccharide, or you could also think of it as a polymer or chain of glucose. So you take individual glucose molecules and string them together to create this larger storage molecule we call glycogen, which can then be stored by the liver until it's triggered to release that for later use. So the bile, again, is being used to break down tougher materials, in particular lipids. Lipids are fats, and if you remember, fats are hydrophobic. They do not like water. They don't mix well with water. And so the introduction of bile and mixing that with the fats emulsifies it. And what that means is the fats are now going to be soluble in water. If we did not have bile helping to break down and dissolve the fats, the fat would pass through your system unmetabolized. We wouldn't break it down into any kind of usable material. Bile is going to be produced in the liver. It's going to be sent out through that porta hepatis, where it will meet up with the duct coming from the gallbladder and direct these down through the pancreas into the duodenum. So while we're talking about bile, we also have to talk about the gallbladder. The gallbladder is this bean-shaped organ that sits directly underneath the liver. It is in fact bright green, I know I've said that before, because bile is green in color, and the gallbladder is literally a storage pouch for excess bile. Now, it's important to remember that the gallbladder does not create any bile on its own. Instead, any excess bile that's created by the liver that is not immediately needed by the body and sent into the duodenum is instead shunted into the gallbladder, and the gallbladder will store that until the individual eats a particularly fatty meal and needs extra bile to break down those lipids. So as far as the bile transport, we have names for each of the portions of this system. We have the hepatic ducts, which are how bile is drained out of the liver. You have the cystic duct, which is the pathway that connects the gallbladder to to meet up with the bile from the liver. And these hepatic ducts are going to meet up with the cystic duct to form the common bile duct. And it's the common bile duct that's going to enter into the duodenum to release that bile into the digestive juices to help break down those lipids. I mentioned this earlier today in this morning's lecture. Gallstones are something that can happen. It's a somewhat common affliction. You can think of them almost like a pearl. It's a very hardened material that can occasionally grow larger over time. Many people have gallstones, but most of the time they are small enough that they can either be passed very easily or they simply remain within the gallbladder, causing no issues. In some cases, though, that pearl-like stone can get larger and larger and eventually become so large that it will block the bile duct either immediately next to the gallbladder or somewhere along this bile duct system. I happened to look this up earlier today. The records for gallbladders are varied, but it seems like some of the largest on record are close to 10 centimeters in diameter, so you can just imagine how large these structures can be. All right, the next ancillary organ that I want to talk about is the pancreas. And the pancreas is a gland, so its job is going to be producing materials like hormones and enzymes to aid in some form or another. In this case, it's producing digestive enzymes that are going to help break down the large organic molecules that we will then be turning into usable materials by the body. It's also going to be releasing those bases to neutralize the stomach acid coming out of the stomach to make sure we're not sending acidic material into the small intestine, because we don't want to be breaking down the lining of the small intestine. And here you can see we have the pancreatic duct. That's how we transport all of the materials being produced by the pancreas, and it's going to exit into the duodenum. The common bile duct is coming down from the gallbladder and the liver, and it is going to meet up with the pancreatic duct. So that's where I meant that the bile ducts are going to pass through the pancreas. It meets up with this pancreatic duct, and then they both empty together into the duodenum. So the pancreas also does several other things for us. In particular, it helps to regulate our blood sugar levels. So we've talked about this before, how glucose is being monitored within your blood to make sure that we have enough to survive, but we don't want too much because that can cause breakdown as well. When blood sugar levels are detected to be low, this will stimulate the pancreas to release the hormone glucagon, which is going to promote the release of glucose into the bloodstream, thereby increasing blood sugar levels. If blood sugar levels are detected to be too high, the pancreas will release the hormone insulin, and the insulin will act to promote the take-up of glucose by liver and other tissues to store it until it is needed at a later time. So we'll talk a little bit about insulin here. I know I covered this when we went through the endocrine system last semester, but in case you weren't in my class, or if it's just something that needs to be refreshed, I'm going to go over it again in a fair amount of detail. So it's going to be produced by the pancreas, and insulin's job is to lower blood glucose levels. And it does so by promoting the uptake of glucose by the cells in the body. The glucose molecules that are taken up by these cells can then be strung together to form that polymer or chain of glucose molecules, and that's what we call glycogen. Now, you may just remember I just mentioned the glycogen a minute ago when I was talking about the liver, and that's because the liver is one of the primary storage places for glycogen. As for the regulation of the release of insulin, whenever you eat food, your body knows that it is getting a new supply of glucose. Therefore, it wants to secrete insulin to make sure that any glucose that's already in the blood can be taken in and stored by the cell tissues. That way we prevent the the glucose levels from skyrocketing because there was already glucose there. We've taken it out and it can be replaced by the new glucose from the food that you're just consuming. Too much insulin can also have issues. It's going to decrease blood glucose levels to a level that is below safe levels. You do need some amount of glucose in your blood. And the big reason for that is your central nervous system, if you recall, is behind that blood-brain barrier, which is going to be very selective about how and what materials get to the brain. And one of the things that passes through is the glucose in the blood. That is the only way that we get glucose to our nervous system, is passing it through the blood-brain barrier. So if the blood glucose levels are too low, you are not getting sufficient amounts of glucose to your central nervous system. And that's going to deprive it of the energy it needs to function, which can lead to various neurological symptoms. Conversely, you can also have excess insulin in the body, so too much production of insulin. And what happens here is your body becomes resistant to the insulin. And so it's not going to respond to the production of insulin and tell the rest of the body to start storing glucose. In order to make sure that your body does store glucose, your pancreas responds by producing even more insulin, which means you become even more less responsive to insulin over time. Type 2 diabetes is a form that suffers from this hyperinsulinemia, the excess insulin production, which is why without proper treatment, type 2 diabetes is very debilitating and degrading over time, because this process is a positive feedback loop. You're going to produce more insulin and become less responsive, so you produce even more insulin, and it just starts to back up the system. All right, back to that connection with the liver. So glucagon is that hormone that is produced by the pancreas, which is going to do the opposite of insulin. Rather than the clarify, the insulin is telling the body to store glucose. It's going to take it out of the bloodstream. Glucagon is going to have the opposite effect. Instead, it's going to tell the body's tissues to release glucose into the body to increase the blood glucose levels. So in our connection to the liver, glucagon is released by the pancreas. It is sent to the liver, and the liver is going to take those glycogen polysaccharides that it has been storing and break them down back into those smaller glucose molecules, and it can release that into the bloodstream, which will result in increasing blood glucose levels. The hormone glucagon is going to have the same effect as it does with the liver in muscle and adipose or fat tissue. So it's not just communicating with the liver. Glucagon is going to be sent out into the bloodstream to reach lots of different types of cells, to tell those cells to release their stored glycogen, break it down into usable glucose to make sure that we build up the concentration of glucose in the blood. So what stimulates the pancreas to release insulin? It's going to be, or the secretion of insulin is going to be promoted by a couple of different things. High blood glucose levels would be an obvious one, because insulin's job is to lower blood glucose levels. So if the body detects hyperglycemia, high blood glucose, it will then stimulate the pancreas to release insulin to lower those glucose levels. There's also the parasympathetic innervation. So the rest and digest system, when you sense that food is going to be coming in, whether you're smelling it, thinking about it, or tasting that food, your sympathetic system is going to trigger the digestive process, which is going to trigger the release of insulin to make room for that new incoming food and source of glucose. There are also hormones from the digestive tract, especially from the duodenum, that are going to tell the pancreas to release insulin to make sure we are aiding in digestion over time. All of these are going to lead to increased insulin following a meal, which is going to help lower blood glucose levels to the safe levels that we want to maintain. Glucagon secretion is going to be following very opposite effects. So low blood glucose is going to trigger the release of glucagon. Sympathetic system is going to trigger the release of glucagon, and so on. Insulin secretion is going to be decreased by low blood sugar levels. So this is called hypoglycemia. So the low blood glucose levels, if detected, are going to decrease the production of insulin, and instead, if I go back, increase the production of glucagon. And then, as I said, sympathetic innervation, which is going to be your fight or flight system, is going to shut down digestion, which is going to decrease the amount of insulin being released. So we've talked about the three main ancillary organs, but there are a few other structures that I want to include here because they do fit within the realm of the digestive system in some way or another. The spleen is one of them. We've talked about the spleen when we talked about the lymphatic system, and truly it is part of the lymphatic system. However, we generally included in the discussion of the digestive system because the blood from the spleen is considered dirty, just like all of the blood that's being pulled from the small and large intestines. And so all of that blood is going to be pushed through the hepatic portal system to be filtered by the liver before being put back into the inferior vena cava. So that's really the only reason we include it, because its blood supply is going to be mixed with the blood supply coming from the digestive system to be filtered by the liver. We have also mentioned the peritoneum, but I want to reiterate this. So the lungs have that sac of membrane called the pleura, the heart has the pericardium, and your abdominal organs have the peritoneum. So this is going to be that fluid-filled sac. You can think of it kind of like a water balloon that you have pushed your fist into. It surrounds your organs, provides a cushion, as well as a form of buoyancy for them, so they're not just sitting on top of each other. What I did not cover, although I did cover when we talked about the pericardium, just to reiterate, the outer membrane has its own name. We call this the parietal portion. This is going to be the portion that is up against the inside of your abdomen. And then we have the visceral peritoneum, and this is the portion that is directly in contact with your organs. Visceral, again, meaning organ. So you can remember that the visceral peritoneum is the portion of the fold that sits in contact with the organs themselves on the inside of the peritoneal sac. We also discussed that your organs, while they're sitting in the peritoneum, are going to need to have access for blood, vasculature, and nerves to reach them and supply them with what they need. And they reach them through these mesenteries, which are folds in the peritoneum that allow, it creates a passageway for vasculature to reach from the back of the abdomen, where it's coming off of the aorta, and passing forward through the peritoneum to reach those suspended organs. Now, there are two additional folds found within the peritoneum that aren't just called mesenteries. Instead, we call these the ommenta. And they have their own names. We have the greater omentum and the lesser omentum. And they are going to act to separate organs and to help hold them in place within your abdomen. In addition to these basic roles, it's also going to be a location for visceral fat deposition. So you're going to act, or the ommenta are places where we store excess fat in our body. We'll talk about the greater omentum first. And the greater omentum stretches from the inferior border of the stomach and crosses the transverse colon. So the transverse colon is the upper portion that crosses horizontally across the body, and then passes to sit across and atop the small intestine and the other portions of the large intestine. Now, it's important to note that the transverse colon is actually inside the greater omentum, so it's kind of enveloped by this fold of tissue. It's a really interesting structure, especially if you've ever had the opportunity to do a human dissection. It's not really attached particularly well to any of the organs sitting underneath it, and so you can physically lift it up, and it will lift the transverse colon up with it because the transverse colon is passing through it. So that's what this figure is showing you here. Someone has taken the greater omentum, lifted it, and folded it up across the thorax, and that has lifted the transverse colon so that you can see what's underneath the greater omentum. The lesser omentum, as the name suggests, is a much smaller structure. It's going to be stretching between the superior side of the stomach over to the liver. And this one serves to contain the portal triad. So those three vessels that I discussed earlier, the blood supply to the liver itself, the dirty blood that's coming in the liver for detoxification, as well as that bile duct taking bile from the gallbladder and the liver and passing it down to the pancreas. All three of those are going to be encased in the fold of the lesser omentum. This figure is particularly vibrant, I would say, but it's trying to illustrate to you this yellow color is the color of fat, and these omentums are going to be places where we can store additional visceral fat in the stomach area. All right, one of the last things I want to talk about on this lecture is gut bacteria. So I've mentioned this before. We have a very diverse and abundant flora of bacteria that live within our digestive tract. Most of these are benign, meaning they will not make you sick as long as they stay exactly where they are supposed to stay, so within their designated region of the digestive system. These gut bacteria are going to be used to aid in digestion. There are some materials that we eat that we are not particularly efficient at breaking down or from extracting nutrients from, and the gut bacteria can do those things, and so they help to break that material down within our intestines, and that allows us to then absorb those nutrients more efficiently. The gut bacteria can also produce byproducts that can be beneficial to us, including certain vitamins. And I mentioned before, one of the big ones is vitamin K, which is a necessary vitamin for blood clotting as well as the production of bone material. And while we can get some vitamin K from our diet, the vast majority of our vitamin K production is actually happening by the gut bacteria in our digestive system, and that's where we absorb that vitamin. Now, we talked about antibiotics when we talked about the immune system, and antibiotics are designed to kill bacteria when you are experiencing an infection, and they're very good at that. The issue is that antibiotics are not selective. They're not just going to take care of the infection that you're experiencing from a cut to your arm. Instead, they are going to target bacteria in a general sense, which means when you are taking antibiotics, you can actually decrease and change the population of bacteria within your digestive tract. So you can decrease those bacteria levels, which over time are going to rebound, so you're going to get bacteria back. Or, interestingly, if someone has taken antibiotics, they can actually see a change in the dominance of bacterial species within their gut tract. So you may have had species A being dominant beforehand, but after the antibiotics after the antibiotic regimen, you can end up with having bacteria D being the dominant species, for example. So that's a response to taking antibiotics. However, we also see the change in our gut bacteria as we age. This is a natural process, which is why something that you may hear family members discuss is, oh, I used to eat this all the time and I never had an issue, and now it's becoming harder for me to digest. I have more reaction to eating this food. It's because as you age, the diversity of the bacteria in your digestive system is changing, and that bacteria may not be quite as efficient at processing this food as it was when you were a younger individual. Another little side note here, for humans, gut bacteria is not strictly necessary. They are very beneficial to us. They help us to break down these tough materials and help us to absorb these nutrients more efficiently, as well as producing those vitamins that we need to function. However, we can get everything we get from the gut bacteria from changing up our diets. We can eat certain things, take vitamins to give us those materials. But some animals, especially grazers that are eating predominantly things like grass and leaves, they cannot live without the gut bacteria. And that's because the plants that they're eating are predominantly made up of cellulose, which is a very tough macromolecule, very difficult to break down and nearly impossible to digest on its own by mammals. So instead, animals like a cow that survives exclusively on this grass material relies on its diverse gut bacteria to break down cellulose to make sure it's getting the nutrients it needs from that structure. As I said, we naturally have bacteria throughout our entire digestive system, and most of the time it's going to be completely benign and not causing us issue in any way. However, you can have abdominal infections, and we typically call these opportunistic infections, where your normal gut bacteria find a way to get into an area where they're not supposed to be, where the body is not prepared to live with that particular bacterium. An example would be the appendix. So you can have bacteria from the colon, which can backtrack and enter into the appendix, which is not used to having that particular species of bacteria in it. And especially if it gets blocked by, for example, food particles coming in from the small intestine, that new bacteria can cause infection. And this is an opportunistic infection. It's just because one bacteria moved to an area it's not supposed to be. And in these cases, it can become dangerous if not treated immediately and can sometimes result in needing surgery. This is also why ruptures of digestive organs are particularly dangerous. I'm sure you've seen it in action movies or true crime type things. You don't want to see belly wounds, because when you rupture one of these digestive organs, it is spilling out all of that bacteria that would normally be benign into that peritoneal space. And that peritoneal space surrounds the entirety of your abdominal system. And so any bacterium that infects the peritoneum can now spread and infect all of the organs within the peritoneum. So that's why a belly wound is particularly grave and dangerous for the individuals. Any infections that involve a rupture of an abdominal organ or a digestive organ needs to be treated very aggressively with antibiotics or other medications. And that's all I have. So as our key topics, what are the ancillary digestive organs? What are their primary functions? The peritoneum, the mesenteries, and the ommenta, what are they doing? Where are they? Where do gut bacteria normally live? What happens when they get to places they are not supposed to? What are they normally doing that is beneficial for us? And then the risks of intra-abdominal infection. Why is that particularly bad for an individual? So hopefully you hear this, but as a reminder, there is no class this Friday. I will try to send out an email. If I forget, I am sorry. Otherwise, have a great spring break, and I will talk to you guys next, I guess, 2 Mondays from now. Have a great week.